So it's my great pleasure to introduce Matt Haber. Matt is a professor in the Department of Philosophy, also the chair of the department at the University of Utah. As you can see, Matt is also affiliated with the School of Biology and the Center for Quantitative Biology. Matt is a philosopher of science, philosopher of biology. He has a specific interest in conceptual issues, philosophical issues connected with systematics. So I think we'll hear a little bit about that, and phylogenetics. He worked on the nature of phylogenetic inference, also about philosophical debates about nomenclature and classification in biology. He also does some ethics, including some research ethics. So in fact, if you have a look at what Matt does, it's extremely diverse. I'm just giving some examples of what he's been doing in the last year. The University of Utah, and more specifically the philosophy department and the group of Matt, philosophy of biology, are members of the Feed and Biomed network that we organize together. And clearly there's a reason for that, which is that, as I suggested before, Matt has done a lot to connect philosophy with biology, and also increasingly with medical doctors, and also other sciences. So we talked yesterday about that, the way philosophers in Utah are connected with all sorts of scientists and medical doctors on campus, and this is an important and big campus. Recently, Matt has published a whole special issue of the journal Philosophy, Theory, and Practice in Biology. Completely free journal online, I recommend. Free submission, free reading. This is very rare, very cool. They do it with some money. They prove that it's very easy to do, in fact. So everyone will tell you it's impossible to do. It's very easy to do, in fact. You just need a little bit of a budget from a university, and it works. Anyway, Matt published this whole special issue on species in the age of discordance, and also in the last 2 years or so he worked on individuality from a point of view which is very much connected with evolution and also systematics and phylogenetic that I mentioned before, especially so he wrote a paper called The Individuality Thesis 3 Ways. And also, to take a different example, in 2015, Matt wrote a paper about mitochondrial diversity and the reversal test in the American Journal of Bioethics. So you see once again that the diversity of topics and interest of Matt is pretty amazing. Matt, thank you so much for being here today and giving this talk on positively misleading errors.
Thank you for having me out. It's been a great week in Bordeaux. I've really enjoyed it. We had a wonderful conference on Monday and Tuesday. Yesterday I had a great discussion with the lab group here on how to embed philosophy of science and why we would want to embed philosophy of science. So I'll start. I'm a doctor of philosophy, same as the gentleman in the image there. My wife is a medical doctor, so she is a primary care physician. Her and her friends tolerate me, but what I'm going to talk about today they actually think is very interesting. And that's why I selected this joke. She has this up on her wall at work, and they all think it's funny and laugh at me. But today what I'm going to talk to you about is a particular kind of error that we can make. So a systematic error of inference, a kind of fallacy. And what I'm going to suggest is that this error, I'll start by talking about where it came from, who discovered it. But when we name kinds of errors, when we name kinds of fallacies and identify them, it helps us recognize them better and understand how to handle them more effectively. And I'll come back to what I mean by that in a moment. So today what I want to do is give you a general account of what kind of error this is. It's called a positively misleading error. So I'll give a general account, a 30,000 foot or 30,000 meter view of what that means. And then I'll start by describing a case that we discovered this from about 40 years ago, and then talk about a potential case in clinical medicine. First, so why philosophy of science? So why am I interested in this? And why am I connecting this to something like clinical medicine? So when I think about what philosophy of science is, I think one of the central driving questions, not the only one, but one of the important driving questions for a philosopher of science, is to try to give an account of what makes good science good science. So why is good science good? And that can take lots of forms. It can be what counts as a good scientific explanation. It could be what's good methodology in science. These are the kinds of things that philosophers of science discuss. It can also be a value or normative claim. Why ought we do this kind of science? What are we doing science for? Why do we value it? In this case, though, I'm talking about what good scientific reasoning looks like. And we can identify, one thing philosophers of science do is identify what good scientific reasoning is, or where good scientific reasoning falls short or goes astray. And this is an example of that. But the fallacy is not one that's very well known. So positively misleading errors, they're well known in phylogenetics, in the area of biology that I'm most interested in and spend most of my time looking at. And they're so well known, if somebody says, that looks like a positively misleading error, everyone knows what that means in the field, and everyone can evaluate that claim and assess whether or not there is a positively misleading error happening or not. That turns out to be really important because it helps them advance the science and understand what's happening in that case. And what I want to argue for today is that if we can, my goal is to spread the word about what positively misleading errors are, so we're better able to identify those in other fields and improve the reasoning and avoid fallacies or bad inferences in those fields. So, I'm going to start by describing an exemplar case. I'm going to give kind of a cartoon example just to help you see what these kinds of errors are. Then I'll talk about certain features that are common to them. I'll generalize a little bit. Then I'll offer a little bit of history about what the response was to the first discovery, because there was a lot of resistance to recognizing these as errors, or the error in particular. And finally, I'll end by discussing a case from medicine that I think is a candidate case, we'll say. I know it's a candidate case. I think it's a good case. So let's start by talking about what these things are. So I'm sure that right here, I probably don't need to go any further. Everybody know what that is? Yeah, nobody knows what that is. So this is from a 1978 paper by Joe Felsenstein called Cases in Which Parsimony and Similarity Methods Will Be Positively Misleading. And that's where the name of this error comes from, positively misleading errors. This is called long branch attraction. I'm not expecting you to remember that or even understand what I'm saying at the moment. I'm just pointing at this thing here, and we'll come back and talk about what that is and why it's significant. Okay, so we can give a general description of what a positively misleading error is. This is a first pass at a definition. So a positively misleading error is a case of scientific reasoning in which adding more data systematically and reliably produces greater strength of support for an erroneous hypothesis over the correct one. What's that mean? That means as you add more data, you can add good data. As you add more data, you get stronger support for the wrong answer. And that is systematic. That is, every time you add more data, you're likely to get higher support for the wrong answer, and reliable. As you keep doing it, you're going to keep getting stronger and stronger support for the wrong answer. It's the exact opposite of what's called statistical consistency. So in statistics, a problem is statistically consistent if you're guaranteed to converge on the right answer as you add more data. Something is statistically inconsistent if it's not statistically consistent. So if you won't converge on the right answer with more data, it's not statistically consistent. Positively misleading errors are a special case of inconsistency, where you're guaranteed to converge on the wrong answer. And it's a nasty problem because it looks like good scientific reasoning, but it's getting you to the wrong place. So that's not great. Okay, so let me give you a toy example to show you the kinds of cases where we discovered this. So down here we've got some sheep. We're going to call those wild sheep. Those are not wild sheep, but we're going to call them wild sheep. So suppose there's a population of sheep living in a valley, all right? Happily munching along the grass and having a good old time, all right? Then this population splits in two, and you get two new species. You get this species and this species here. They still live in the valley, so you've got two new species living in the valley. You can even suppose this one went extinct or got replaced or transformed into one of those. Doesn't really matter. So you started with a valley population, they split into two different species. Soon after that, these species also split into two. And for each of those valley species, one of the splits goes up into a mountain and the other goes up into a mountain on the other side. All right? So we had a population of sheep split into two new species, both of which subsequently split into two new species. In each case, one stayed in the valley, one went up into the mountains. Okay? All right. So, now there's relative stasis. There's no more splitting, no more new species. And these different populations, or these different species, they live in the valley or they live up on top of the mountains for a really long time. And while that's happening, they're transforming. And they're transforming in response to ecological and environmental pressures. All right? So the valley species, they still kind of resemble the ancestral population. But the mountain species, we've seen a lot of change happening there because they're adapting to living up in a mountain. So we see physiological changes, we see morphological changes, all kinds of changes happening. Right? So they're living, these two species, even though they're not as closely related to each other as they are to their sister species down here, they resemble each other because they've been transforming in response to similar pressures. So let's clean this up a little bit. So I got rid of pictures of the ancestral species and I connected these lines. So this now looks like a phylogenetic tree or an evolutionary tree. And what you see here, there's a few things to notice. These lines, the way I've drawn this, the lines correspond to how much change we might expect to see over time. Okay? So the branch length, these are called branches, branch length corresponds to change. So that's a measure there. Also notice that this valley species is most closely related to this mountain species. So each mountain species' closest ancestor is a valley species. Okay? All right. Suppose now you are a biologist and you are a sheep biologist and you want to learn about these species. Well, one of the first things you're going to do is you're going to want to reconstruct the evolutionary history of these species, because that's going to help you understand and explain the biodiversity that you see, among other things. But you can't see the past, right? None of us can see the past, not directly anyways. All you can see are these living species. So you have 4 species living in the valley and mountains, and you want to reconstruct that evolutionary history. Well, what method or will you employ to do that? Well, one you might use is you might just compare the different species and ask which ones are most similar to each other. And you might hypothesize, I'm going to construct a tree that offers the simplest explanation for how to reconstruct this history. And I want to do that in a way that requires the fewest number of changes over time in the entire population. So you might do a similarity tree. If you do that, here's what you're going to get. You're going to lump together the 2 mountain species because they're going to be most morphologically similar. And you're going to lump together the 2 valley species. The problem is that's wrong. That's not what the actual evolutionary history looks like. But you're using the wrong model to reconstruct that history. And it doesn't matter how much more data you add. You can add as much morphological data as you want, physiological data. It even turns out, if you're not careful about it, even looking at molecular data, all of those are going to support this tree more and more and more. Okay? So that's called long branch attraction, is the name of this problem. It's an example of a positively misleading error. As you add more data into this study, you're going to get stronger and stronger support for the wrong hypothesis. And it's a really nasty epistemic trap. It traps you in the wrong epistemic position because it looks like good scientific reasoning. You're going out, you're collecting good data, you're plugging into your model, you're looking at what hypothesis it supports, and it gives you support for the wrong thing. This is what was described by Joe Felsenstein in this 1978 paper. This is the model he used to describe that. So this graph here looks at the relative lengths you need on different trees. So we have Q's here. These are the length of those branches, and P's here. And he looked to see under what conditions are you guaranteed to get the right answer or wrong answer. And he described a situation where if you have long branches that are connected to each other by a short branch and have short branches as their most closely related species, you're guaranteed to get the wrong answer when the relative length of those branches is in that area. So that's a positively misleading error. That's called long branch attraction. It's a really famous case in systematics. What I'm going to do now is kind of talk about more general features of that and then what the response was to this. But this is a general feature that I want to describe. So long branch attraction was the first time the phrase positively misleading got attached to this. And this is what Felsenstein named a positively misleading error. Okay, let's talk about some general features of positively misleading errors. So I want to focus on three kind of general features we see of these kinds of errors. And then we'll talk about why they're so hard to dislodge. Because it turned out, much as this was a great paper, people are still arguing about this. And it's still, people are so skeptical about this as a problem. Not as many as once were. But it took a long time and a lot of work to convince people that this was a real thing. So let's talk about why that might be. So first, what's it mean that they are positively misleading errors? I want to explain that this is a statistical problem, but it's a very unique and distinctive statistical problem. Right? It's a feature of the way that our statistics are working when we have a model that's being oversimplified or we're tracking the wrong outcomes or measures of something. Okay? So sometimes when I give this talk or people read about this, they wonder if it's just a type 1 error or a false positive. It's neither one of those. So let me explain why. So a type 1 error occurs when you're doing statistical testing of hypotheses. And along with type 2 errors, describes how the role of chance might be misattributed in trying to explain a pattern in the data. So one way to think about it is it's a mismatch. So type 1 and type 2 errors are mismatches between the data set you sampled and the population at large that you're trying to draw an inference about. All right? So if the sampled data set displays a particular pattern, we can ask whether that pattern, whether it reflects or whether it displays that pattern because it's reflecting the general population at large, or whether you just got unlucky and you picked the wrong stuff. Right? So if you want to study a population of 10,000 and you randomly pick 100 units out of that population, there's some chance that the units you pick won't have the same patterns that you're looking for. And whether you attribute the pattern you see to that population by accident or whether you fail to is a type 1 or type 2 error. Okay. The way you resolve a type 1 or type 2 error is you go get more data. Right? So if you are worried about having a type 1 or type 2 error, you can resample and you can generate more data. And the more data you get, the more likely you are that your data set will resemble the population at large, and you can reduce that level of chance you're going to get it wrong. Right? Sometimes this gets expressed as a p-value, for example. Positively misleading errors, though, don't get resolved that way. Right? Because you can sample more data, but you're not going to resolve the problem. And the problem isn't that there's a mismatch of data and population. The problem is you're expressing the relationship of the data set you're looking at incorrectly to the population as a whole. Okay. All right. Another feature of positively misleading errors, at least long branch attraction, is you can actually take the data sets you used that generated the positively misleading error, and if you run it with a better analysis, you'll resolve the problem that way. That also suggests it's not a problem of sampling or chance. So for example, with long branch attraction, it's a problem for certain methods in phylogenetics. But if you use other methods, for example, you use Bayesian or likelihood analyses that incorporate certain models of evolution, you can resolve and discover those problems. So it's not just it's not a matter of having a bad data set. It's also not a false positive or false negative kind of problem. Those are functions of low specificity or sensitivity, which are respectively the ability to correctly identify positive and negative results. Right? So in other words, it's not that we're wrongly calling a nucleotide an A instead of a T. Right? So we're not looking at the data incorrectly or reading it incorrectly. Those are problems of false negatives or false positives. Again, those can be resolved by resampling, making sure our testing equipment's working correctly, those kind of things. But these are good data sets. They're big data sets, and adding more data doesn't resolve them. So positively misleading error is distinct from other kinds of problems in statistics. All right. These generate epistemic traps. I mentioned this earlier, but I want to really come back to this and really emphasize it. It mirrors at least one good form of scientific reasoning. Now, I happen to think there's lots of good methods for reasoning in science, lots of good ways to make good inferences in science. And this mirrors one of those, right? Who doesn't want to have a great data set that you can grow with more good data and get increasingly strong signals supporting the hypothesis that looks really intuitive? That sounds great, right? We do that. That's what we want to do in many cases. That's exactly what's happening with positively misleading errors and why they're so difficult to dislodge when you run into one. If you've studied philosophy, and I'll show a picture of this in a little, or of this person a little bit. If you've studied philosophy of science, odds are you've probably studied Karl Popper and his theory of philosophy of science that we should be trying to falsify our hypotheses. So another response I sometimes get is, well, isn't this just a problem of confirmation, right? It's not good to do science just trying to prove something, because if you go out looking for evidence confirming something that you want to confirm, you will almost certainly find that evidence. That's not good science either. This is not a problem of confirmation, though. It's a problem of convergence. And in fact, what Felsenstein was examining was a method of science based on Popper's theory of science of falsificationism. So he was looking at cases where people were testing phylogenetic hypotheses, and the more tests that it survived, the stronger we took those hypotheses to be corroborated. Right? So it was a falsificationist framework that he tested. Now it's not clear that it was actually embodying falsificationism, but it's probably about as close as you'll find in science. The problem wasn't that it was confirming it, that it was converging. So there was stronger and stronger support that these hypotheses were being corroborated, that they were passing harder and harder tests of phylogenetics. So any method that relies on convergence is susceptible to this. And that's especially true if the entrenched commitments are really intuitive and if it's making sense given what we think is happening in a system, right? We use our intuitions. We look at systems and we often will step back and ask, do these results make sense? Is this fitting with the other things that we believe about this system? That's part of what's happening in these cases. So one of the challenges is that long branch attraction, this case here, it disrupted a lot of entrenched commitments about phylogenetics. I don't want to give a whole history of phylogenetics here. I mean, I would love to give a whole history of phylogenetics, but I'm just going to give a really, really quick and brief history. So in 1978, phylogenetics was coming out of a period where there were lots of fights about what good science was in evolutionary biology, and in systematics in particular. And one of those big fights was, well, we're looking at a single historical event. Are we able to reconstruct a single event in any kind of philosophically and scientifically justified way? And a lot of the fights in systematics at that time were over whether there was a good scientific method for reconstructing evolutionary history, or whether the best we could do is just look for clusters of similarity. And a really important conceptual move that happened was people who were reconstructing histories used that falsificationist framework to argue we're not merely using circular reasoning here. We're testing these hypotheses in a scientifically responsible way. So one of the challenges that long branch attraction disrupted was this commitment about what testing hypotheses meant and whether this was a good scientific method. What Felsenstein was arguing is that this should be viewed as a kind of statistical problem that statistical tools could be used to evaluate. That challenged a lot of people's research programs, and not just their research programs, but their justification for whether what they were doing was science or not. Okay, so I'm kind of hinting at the next section, which is, what was the response from the scientific community to Felsenstein's discovery? This is a really cool discovery. If you read this paper, it's a very short paper, and it's a really clean, beautiful paper. And beautiful is one of those terms in science. It's hard to kind of capture, but it's a really elegant proof. The math isn't that easy, but it's not that hard. It's this elegant proof that's really convincing. So what was the response? Well, wasn't great. So I want to talk about three kinds of responses. Because, long story short, his discovery was not warmly embraced. And the debates that followed were really, really heated. So anyone who's ever looked at the history of phylogenetics or history of systematics will know that there were lots of really angry, hard fights in that field. I was at lunch, I was talking a little bit about this. People would sit on opposite sides of the room at conferences and yelling matches would ensue between them. It became personal, became really hard debates. And this went right along with that. So this split the community in a lot of ways, and there was all kinds of fights. So I want to talk about three big kinds of responses. The first was, before I go on, I want to mention one thing. The reason that I'm mentioning this here isn't just because it's kind of fun gossip, although I guess it is kind of fun gossip. But the reason I mention it is because these kinds of errors are going to often be challenging, entrenched, intuitive commitments. And the kind of response that we see to these are likely to be the kinds of responses we might see when someone identifies something as a positively misleading error. And my hope, I'm a hopeful guy, my hope is that if this kind of error is better known, rather than generating these heated kind of responses, instead we can evaluate whether in fact there is a positively misleading error occurring there or not. So it'll redirect some of that reaction to assessing whether something's genuinely a positively misleading error. So, the first objection was philosophical. Here's Karl Popper. I already mentioned this a bit, right? It was disruptive because the technique that was being challenged, parsimony, wasn't just a technique, but it was a central identifying commitment to how we do science in this area. And so challenging that commitment, challenging that method, challenged the very notion of whether, on some people's view, challenged the very notion of whether you could even do good science in this area. Now, it was also a productive disruption. So I'm not really focusing on that part too much in this talk, but I do want to mention that a lot of these disruptions ended up being productive, and that they were moving the science forward in particular ways. So even though this was a really bitter debate, people like to argue about philosophy. They get really heated about it. But in this case, it was productive in two ways. Proponents of the parsimony method, proponents of falsificationism, they had to do a better job of articulating and specifying precisely what they meant by falsificationism. And if you are a Popper scholar, there's this whole literature in the systematics world on what Popper might have meant. And much as I'm not a Popperian and I'm not a falsificationist, it really forced them to be more careful about what they meant in that context and to try to articulate how those methods they're using were formulated to embody this falsificationist, this testing hypothesis approach. It also prompted people who were proponents of Felsenstein's approach, the statistical approach, it prompted them to offer a justification for using statistical analyses for cases like that, which, as we'll see in just a moment, also help them develop statistical tools beyond just phylogenetics, but tools that got pulled and used in lots of other areas as well. And to think about what statistical reasoning meant and why and where it could be used. Okay. Another kind of response was less confrontational, but still challenged it as well. And this was a response that said, well, even if long branch attraction is correct, let's suppose that Felsenstein was right, his paper is very abstract, right? It doesn't look at an actual case in biology. It looks at cases of the way trees could possibly have unfolded and evolved. And so this challenge was, well, okay, long branch attraction is a real thing. That would be a positively misleading error. But how common is this in nature? We need empirical cases, and it's an empirical question how much of a problem this actually is. And one of the motivations for that is that the methods that Felsenstein challenged, they're computationally inexpensive. Whereas the methods he was arguing we ought to use instead cost a lot more computationally. They just take a lot longer to use, and they're a lot more complicated to write and construct. So there's an advantage in being able to use these other kinds of methods. So this was disruptive in a particular way because it meant leaving aside, again, this philosophical commitment to falsificationism and moving towards something like statistical analyses. But it was hugely fruitful because we end up getting lots of studies that first are looking for other cases of long branch attraction, and we found a lot of those now. So fruit flies are an example. This is an example, I believe, in trees, pine trees maybe, I don't know. So there's lots of examples that we found that look like they're good cases of long branch attraction. But in doing that, we also discovered empirical conditions under which lots of different methods perform better. So a lot of the work that happened in phylogenetics in the ensuing years, in the 80s and 90s, was trying to figure out what kinds of statistical methods are appropriate for different kinds of conditions. And this brings me to the last point, adopting the statistical approach. This was a third reaction. So some people adopted Felsenstein's approach, recognized that statistical analyses are really useful in this context, and got to work trying to develop lots of statistical tools. Now this is great for phylogenetics, and I'm sure you all are as enthralled and excited about phylogenetics as I am, but it's better than that too. Because phylogenetics was one of those sciences that was on the early end of the big data boom. So 40 years ago, it's not like we're sequencing genomics back then, but there's still a lot of data that systematists could work with. And so they've been using these really massive data sets. And it's not just in terms of how much data there is, but the data is very complicated and complex. And what I mean by that is that the way the data relate to each other is itself as interesting as what the patterns are that we can find in those data. And so the models they started building are tools that we can now use to navigate through these really complex systems of data. And that has real benefits for the rest of science, as the rest of science is now wrestling with these big data challenges. How do we work with big data sets when the individual parts of those data are connected and causing each other in really complicated ways? Okay. So let me conclude by posing what I think is a candidate case outside of phylogenetics, because no one cares about phylogenetics with me. I understand that. So I want to look at a candidate case in medicine, right? And this is what my wife and her friends got excited about, and they think there's all kinds of errors like this in medicine, and they gave me this case, so we'll see what happens. All right, so I think there's almost certainly going to be positively misleading errors in other kinds of sciences. That's part of what I'm looking for, are other places where positively misleading errors are occurring. The thing is, the only scientists who use this name, this phrase, are phylogeneticists. It's them and a handful of philosophers who care about this. But I want to see positively misleading errors be as well understood and as familiar as type 1 errors or false positives. Because if someone says that's a type 1 error, no one gets all freaked out that you're challenging the, oh, you're now saying I don't do good science. Instead, you go and look and you figure it out. And we know what those kinds of errors are, and we know the tools to use to analyze and assess that. That's what I want to see happen with positively misleading errors. I think there's almost certainly some cases in the social sciences where there's statistical examples, but I also think there's more informal ways that this can happen. And I'm sure some of you can start thinking about all the ways people make bad decisions or bad inferences that look like positively misleading errors, although a bit less formal. I want to look at treatment protocols and standards of care in clinical medicine. So where in phylogenetics, methods generate positively misleading results, in clinical medicine, I want to suggest that we can get standard treatments or standard protocol for treatments that are generating positively misleading responses to certain kinds of conditions. Okay, so the example I'm going to talk about is septic lactic acidosis, septic shock, right? So septic shock is really, really bad, all right? So what happens when you go into septic shock? Septic shock means your blood is becoming more acidic. That's bad, and it's bad for all the reasons that you're thinking it's bad and more. So here's one reason why it's really bad if your blood starts becoming acidic. The proteins in your blood, you want your proteins to do stuff in your blood. If you start lowering the pH, guess what happens to your proteins? They start getting denatured and doing weird things, and then you die in many cases. This isn't good. So, for a long time, what was the standard treatment? Well, the standard treatment was, your blood's getting acidic, let's add a base to your blood to raise your pH back up. We're going to use sodium bicarbonate to do that. Baking soda is what we call them in the US. I don't know what the phrase is in French. Sodium bicarbonate is a base. If you put a base into an acid, it'll start raising that pH back up, right? And doing that helped people. People did better. So if someone's going into shock and you do nothing, they don't do well. If you give them sodium bicarbonate, they are more likely to do well than they otherwise would. Okay? So patients receiving this intervention, they did better. And we have a lot of data showing that. But the problem is, that's probably a lousy thing to do to someone. Now, it's good to help them, but you don't want to give them sodium bicarbonate. This is a paper from 2000 in Chest, which is a really influential journal in medicine. And what they argue is that sodium bicarbonate, sodium bicarbonate is generating a positively misleading result. Now, they don't use that phrase, but that's what they're doing. Okay? So, methods are positively misleading in phylogenetics, and they converge on an erroneous hypothesis at the expense of a better one. In clinical medicine, it's treatment protocols or standards of care that can be positively misleading, but they converge on the wrong treatment at the expense of better ones. And that's what their argument states. So I'm going to quickly go through that just to kind of show you that similarity. Okay? It's unfortunate that they didn't have the term positively misleading error and that statistical tool to use to describe what was happening. So one way, my advisor used to talk about philosophers of science filling a bookshelf that scientists or other philosophers could pull tools off of to use when they're doing science. And it's really unfortunate they didn't have the tool of positive misleading errors, because that would have helped make their case. Okay, so what's positively misleading about this? Well, I'm going to quickly just give you the structure of their argument and what some of the responses were. So their strategy follows a pattern that's really similar to Felsenstein's. What they do is they identify 4 core assumptions that justify the interpretation of evidence as increasingly supporting the entrenched treatment protocol. Right? So these are the 4 assumptions or points that they identify people are using to justify administering sodium bicarbonate. Okay? And what they do, they proceed to go through each one of these points. I'm not going to go through each one of those points. but they proceed to go through each one of those and show that each one oversimplifies what we know about physiology. And that it's the oversimplification that's distorting the way we're interpreting the data and interpreting the model that we're using to evaluate this treatment protocol. So, for example, it sure makes sense that sodium bicarbonate would increase pH. It's true. Sodium bicarbonate will increase pH. It's a base. It raises pH. But that assumes an overly simplified notion of physiology. So in the body, it might raise pH in the blood, but doing so might happen at the expense of what's called the intracellular pH. And it's the intracellular pH that can actually be harmful. So sodium bicarbonate can actually lower the pH in the place where we want to raise the pH. So each one of these cases, they go through and they kind of explain how a more realistic and a more complex notion of physiology tells us that sodium bicarbonate isn't just doing nothing, but actually could be harming the patients. Okay. So why are people doing better? Right? Why are we getting these positively misleading results? Well, it turns out there is a way to help people who are going into septic shock, and it's instead of giving them sodium bicarbonate, you just administer saline, basically water. And the problem is, when people would administer sodium bicarbonate, they do this in a saline solution. So the saline was helping people get better more than the sodium bicarbonate was harming them. So all this data that we saw, right, just like in the other cases, we don't need new data sets. We go back and we re-examine the old data sets with a more complex, realistic model. We can see where that pattern was coming from that was giving that false support for the erroneous treatment. The other thing that makes me think this is a good case for a positively misleading error is that it was really hard to dislodge this treatment protocol. So sodium bicarbonate, I'm pretty sure no one's teaching this now. And maybe there are doctors in here who can tell me otherwise, but I'm pretty sure treating septic shock with sodium bicarbonate is no longer considered the standard treatment. I think in some places it's still accepted. If some old doctor wants to do it, nobody will really yell at them, but I think that's changing too. But this wasn't discovered in 2000. I mean, Forsythe and Schmidt wrote this paper in 2000. This was really influential. This helped dislodge that treatment. It was an important paper, although it still took a while from this. But people knew that sodium bicarbonate wasn't working as early as 1930. So almost 100 years ago, we already knew that sodium bicarbonate was misleading us into thinking it was helping. I don't know the exact details of how we discovered it. My guess is someone forgot to put sodium bicarbonate into the solution, and someone did just fine, and probably started studying it from that. But because it's so intuitive, right? Someone's pH, their blood pH is dropping. Let's get that back up with sodium bicarbonate. And because we kept seeing people doing better, we ended up getting what I think is a positively misleading error in this case. Okay, so in conclusion, I think we need to have a better sense of what good scientific reasoning is. I think an example of a fallacy in statistical reasoning, which we use in science, is a positively misleading error. Naming and identifying and understanding these help us better assess when we think those kinds of errors have occurred. Okay, thank you very much. Happy to take questions.
Thank you very much, Matt. I'm going to hear questions. Yes.
Yeah, I have a question. For the first example in phylogeny,
Yeah.
if I got it right, I get the impression that what caused the error is that the parsimonious explanation wasn't true.
Yeah,
Because
that's
it seems to me that assuming that similarity reflects phylogeny, or phylogeny with proximity, it's the parsimonious explanation.
Yeah.
And so the data was consistent to that. And if people keep working on the same hypothesis and collecting data for the same hypothesis, you're going to get the same result.
That's right.
So is that something that could be general for the But I'm not sure that's
Yeah.
the case for the second example.
Yeah, so the question is what generalizes, right?
Yeah.
And so in the first case, the problem is that they're looking for parsimonious explanations of evolution, and they're assuming that evolution will proceed parsimoniously along some particular measure. And instead, it turns out that in some sense, evolution is more complex than that. But in other ways, I think what's happening is they're counting the wrong thing. And so they're counting the number of evolutionary events and looking for the most parsimonious explanation in terms of number of events, when what they could do instead, and what some of these other statistical models do, is look for more parsimonious models of evolution. So how often should we expect changes to occur? So a more parsimonious model might actually lead to more evolutionary changes, but it's still a simpler model of evolution. If we try to explain a parsimonious in terms of number of events with a model, we might need a more complicated model. Now that's not to say that a simpler model is always better though. Sometimes we need more complex models also. So I think what generalizes is an oversimplified notion of what the underlying causal structure is. And that's what we see in that second case also, is an oversimplified notion of what the physiology is, which leads to us counting the wrong thing.
Yeah, that I agree. There's
Yeah.
one control missing in that experiment because you have two parameters.
That's right.
Only one is being controlled for.
Yeah, so if you end up counting the wrong outcome, or you're using the wrong proxy for what you want to be looking for, that can lead to a positively misleading error. So trying to see whether we're lowering intravenous pH rather than measuring, say, mortality outcomes, we're looking at the wrong thing that's giving us the wrong pattern to track. So I think something like that, but that's the real trick, is what is it that we're generalizing? Because it can't just be, well, parsimony doesn't work. It's got to be something else that we're actually tracking in this case, or generalizing in this case. Yes, it's a good question.
Very cool. Thanks, man. One very interesting. So some related to the previous question, when I was trying to wrap my head around what the phylogenetics case has in common with the other case of
Yeah.
the sodium bicarbonate, has it something to do with the way that in the phylogenetics case it was the short path length in the beginning, whereas in the administration of sodium bicarbonate, it's almost inseparable to administer at the same time the solvent solution. So that's something that's
Yeah.
closely linked at an early stage of intervention or of the process that will then have consequences that are very hard to
Yeah.
disentangle statistically once you look at the data or the phenotypic outcome of the intervention
Yeah.
later on.
So I didn't really get into those details, but that's a really great observation, because that's what I mean when I talk about big data, people throw that word around. But big data doesn't just mean that we have lots of it. It means that the parts of those data sets are interacting in really complex ways. And so that would be the way I kind of generalize that, is that in the long branch attraction case, you have a lineage split and then split quickly again. But then every downstream part is related to the earlier parts. And so you've got this complex relation between the different bits of your data. In the septic lactic acidosis case, there's something similar happening where because the sodium bicarbonate was being administered in saline and we were only looking at some features but not others, we're not really recognizing the complexity of those data sets, and it's leading us to measure the wrong things or track the wrong items. So something like that. I think part of what's going on here is recognizing when we have these complex data sets. And in medicine, I mean, a living body is hugely complex. And when we measure it, we're looking for easy things to measure, but sometimes that can lead us astray if we're not recognizing the way that the parts are interacting with one another.
Thanks.
Christopher?
Yeah, thanks. So, very interesting. I had a question the whole way along, and then it
Okay.
turned into two questions with your clinical example. Because the clinical example is kind of interesting because the first reaction is, well, this wasn't properly put to trial according to clinical standards, which would be, well, we give and then the control group
Yeah.
gets the vehicle on its own.
Yeah.
And then you're telling us, well, the vehicle on its own is what does it. So a proper
Right.
clinical trial would have shown that almost immediately,
Yeah.
surely.
So let's talk, so clinical trials. You know, one thing I didn't mention was a rise of evidence-based medicine and the attempts to bring those kind of scientific standards of reasoning and experimentation to clinical medicine. So you're right, if this, I mean, but this is a really old treatment, right? I mean, at least 100 years ago, they're doing this kind of stuff. I'm not sure that the same kind of standards were in place then. And that's part of what Forsyth and Schmidt are arguing for here, is that we should be bringing this evidence-based medicine critical analysis to all the kinds of treatments we're doing. I don't know if that's realistic to expect, but it's probably a good goal. There's also questions of, and this is my area of expertise, but there's ethical questions about how we do those kinds of clinical studies. We need to be careful we're giving people good care and good treatment in those different situations. And I think that complicates how we assess and evaluate these kind of things too.
Because then, so the original question was, so here we have a hypothesis and it's null hypothesis which aren't being properly tested.
Right.
The original question about the first one
Yeah.
was about Platt's paper about strong inference,
Uh-huh.
where he was criticizing already in the 60s the idea of only testing a hypothesis against
Yeah.
null version,
Yeah.
and saying that what was needed was alternative hypotheses to test. So
Yep.
the question is, how do PMEs stand up to strong inference approach?
Yeah, that's a good question. I mean, in a lot of ways, that's what Felsenstein's pushing also, is he's pushing for a comparative approach to phylogenetics. And that what we end up discovering is that there's no one single method that works in phylogenetics. In fact, to figure out which method you need to use lots of methods and look to see where they're conflicting and where they're in alignment, because that ends up telling you what the underlying conditions are and whether you can use this method or this method. I don't know how you translate that into critical care very well, but I agree that I think Felsenstein's right in pushing for that as that comparative approach. You don't want to just look at them in isolation. So I don't know if that addresses the question.
Yeah, and then finally, sorry if I can, you said then that in this PME isn't a type of confirmation bias. The other thing that makes it interesting then with Platt and strong inference is that he was saying you need to do this method, so you force yourself to come up with alternative hypotheses, and then you confront with the hypotheses that you want. So the reason to do that was precisely to combat confirmation bias.
Good. Okay.
So
Yeah.
there's an
I'll have to look
interesting
back at it. That's a nice observation.
Just a question of curiosity. So you said that this example of the septic lactic acidosis was just suggested by your wife and colleagues, right?
Yeah,
So
that's right.
can you say more about this? It was
Oh,
like
sure.
a pure chance, like something is mentioned and you're like, oh, this is like a PME? Can you say
Well,
more about that?
So sometimes if I'm hanging out with my wife and her friends, they all think it's really funny that I'm a philosopher, because they're medical doctors and they love it, but they think, oh, you're this esoteric person floating off in wherever you are. So I started telling them about what causally misleading errors are, and that I was looking for other cases, and I suspected that there'd probably be cases in medicine. And as soon as I said it, a friend of mine who's a pulmonary care physician, so he takes care of people who are in intensive care units, he started telling me about this case, and that I want to be careful how I say it. He said it's no longer standard to teach people to administer sodium bicarbonate at Utah, but it was shocking how recently it was still standard, and that in fact this was the big fight in their field, was trying to eradicate this treatment. And it was still happening recently enough, and he couldn't figure out why it was so hard to convince people that it was the wrong treatment. And so he shared some papers with me, because both of us thought that this had some features that reminded us of other cases that I was looking at, where what looked like really clear results were not being picked up by practitioners in the field. And so the big picture, one thing I'm interested in is, how do scientists shift from one set of commitments to another? And why do they fail to move from one set of commitments to another when there's really good reasons to do that? So that was part of the conversation that prompted it, was his own frustration at seeing people still using this as recently as 10 years ago.
Okay. And a follow-up question, if I may. You described in the first case that there were some responses, some quite elaborate, in the sense that people resisted,
Yeah.
and they had arguments, maybe not
Yeah.
good arguments, but they had arguments. They wrote these arguments in papers, maybe,
Yep.
or in that case,
Yeah.
it's more like a practical
thing, where people are very slow to adopt something for which there's no good, counter arguments against. You see what I mean?
Yeah.
Oh, okay.
So actually, this goes back to earlier question. So one reason, from what I understand, this isn't quite written down in the same way. The more recent resistance, in part, comes to a resistance against evidence-based medicine. And I should be clear, I'm not a huge advocate of evidence-based medicine, but part of the idea behind evidence-based medicine is going back and reassessing a lot of these protocols we use using more scientific methods. But, and I think there's a legitimate criticism that you sometimes lose you fail the value experience in the same way as you do when you're using that method. And so some of the pushback against studies like these were people who were pushing back against evidence-based medicine. And there's some of that, from what I understand, took the form of, look, I've been doing this a long time, I know what works, I know my experience, the experience of my colleagues and the people who taught me, this works if you, and then there's a fear too. If you don't use sodium bicarbonate, you're going to kill people. And if that's going to be irresponsible if that happens to you. And people still die even when they get good treatment for septic shock. And so if you don't use sodium bicarbonate, your patient dies, you've put yourself at risk because you haven't used that standard protocol. And so if you're in an environment where people are resistant to evidence-based medicine to begin with, and that happens, then there's a real risk that you put yourself in a position that you might not have a job, or you might be held responsible for this. So, yeah, I think there's a lot going on there. Yes, Paul?
So I have a general question.
Yes.
As you mentioned, tracking errors can be time-consuming for researchers, for
Yes.
computers.
Yeah.
And I was wondering, is there some reflection on whether there's an acceptable rate of error in science?
Oh, good. Yeah. So how?
And that science is an approximation to reality rather
Yeah.
than getting something absolute and definitive.
Oh, I love it. That's a great question. So is there an acceptable rate of error? And I think we also might ask, is there a desirable rate of error, right? So I don't know what the acceptable rate is. We put p-values on things and we say 0.95, and that sounds great. I sometimes wonder though if we actually want errors to happen, because when things go wrong and we have to try and figure out what went wrong, we learn a lot by that happening. So that's a great question, because that's what this shows. What this shows is, given the relative length of these branches versus these branches, everything down here is okay, everything up here is wrong. And depending on the method you're using, this curve can move. And that really becomes a question of what's an acceptable rate of error, what's the trade-off for the rate of error we might run into versus the ease of programs or methods that we're using, given that some methods are a lot faster and a lot better at kind of spitting out an approximation of an answer. And that was part of what was being debated. The other question was, related to that was, well, if these are very rare cases in nature, then maybe we just shouldn't care that much about those, and we can, after the fact, go back. Because we have so many things we need to do anyways. If we get stuck on those very rare cases, maybe it's not worth it. So yeah, that's a great question. I wish I had a better answer, but I don't know. Elena?
I had a question maybe a bit into that too, about if you had any examples in experimental science.
Yeah, good,
where
perfect.
It would be different from the phylogenetics in the way that you can directly manipulate the material.
Yeah.
And also different from the medical case,
Yeah.
where you have possibility of having more like internal controls.
Yeah, okay, good. Well, I'm actually going to give you a case from phylogenetics, because that's the only place they use the term positively misleading error. But one way this got tested was in the 90s. So alongside this whole thing, as this is happening, or concurrently, you get the rise of desktop computing. And desktop computing gets more and more powerful. And so when Felsenstein is doing this, there's not a lot of resources for anyone to go do this. He happens to be on a campus where he had access to resources where he could run these analyses in pretty powerful ways. In the 1990s, desktop computers are powerful enough to do this stuff. And so the biologist David Hillis has these really wonderful experiments where he tests both empirically and using simulation studies whether this is right. So what he does is he gets bacteriophages, and he starts collecting them in agar plates, and then just, you know, as they grow, he puts them in new plates and puts them in new plates. So he generates an empirical phylogeny that he knows what that is. So he actually can now observe the history of that evolution. And then he can use that to back test methods. So, and he can manipulate that also to find places where there in fact are long branches or not. And he can then use that to test whether methods do well or not in those cases. And he's doing that alongside some simulation studies as well. So I don't know cases outside of phylogenetics, because I don't know other cases outside of phylogenetics where they describe positive and misleading errors. I think there's some cases in economics where this happens. I actually think in sports economics, so I'm a big baseball fan in the U.S., right? And if anyone ever, there's a famous movie called Moneyball, which is, I don't know if anyone's seen this game. But what ends up happening is a few economists start figuring out that you're measuring the wrong thing in sports, and you can exploit that. So that might be another case, although I haven't really looked at it carefully enough.
I might have another cool case,
Oh, good.
but frightening one.
In medicine, they always are.
Yeah, screening programs, because there is this idea,
Oh,
and
yeah,
we discussed that 2 days ago actually.
Yeah.
There is this case of, for instance, cancer. When you screen for cancer,
Yeah.
the idea is that the earlier you diagnose cancer, the better the outcome,
Yeah.
which is true. Generally speaking, it's true.
Yeah.
But the thing is that at the same time, when you diagnose early, you have more uncertainty, because the kind of states you would diagnose are undecided. You don't know whether they will evolve into a full cancer or not,
Yeah.
or never. So the thing is, of course, you will treat a lot of people, and the thing is, so it's not a positively misleading error so far, but it becomes one because the kind of results you get from that is that your rate of survival will increase dramatically, because you will treat a lot of people that don't have cancer.
Right.
So of course they will survive.
Yeah.
And the thing is that the more you do that, and the more you tend to say, well, we should treat people earlier and
Yeah.
earlier on, because we will save more people.
Yeah, because this population has a higher rate of survival.
Absolutely. Yeah.
Because you're stacking the deck with people who don't have cancer.
Yes.
Yeah.
So I don't know.
That's a
Would
great.
you
That's a
take?
good.
Yeah. Would you take that as a positively
Yeah.
misleading error?
That's a really good example. You have to look at that. That sounds like it could be a good case.
Mm.
You know, a case where knowing what a positively misleading error is would help us evaluate the data that we're generating.
Maybe. Yeah.
To correct for that if in fact it's happening. And actually mentioning the screening, yesterday I mentioned my colleague who's a computer scientist who works on prejudice and bias and algorithms,
Mm.
and he looks at algorithms that are screening applicants for jobs. I wonder if there might be some features in that one also. I'm
Yeah.
speculating on that a little bit. So that's a good case.
Mm.
Yeah, thanks.
One last question, Sophie?
Maybe a stupid one. I wonder if rumors could be socially enemy.
What is that?
Rumors.
Oh, rumors. I started working on this before our current political situation in the U.S. occurred. But I wonder if rumors and some of the more systematic studies of how false news stories get planted and then spread, I wonder if there's some feature of positively misleading error that people are exploiting without knowing what they're exploiting. I think that's very much a case, and that was actually the original case I was trying to look at, that I think would be a good case of informal positively misleading errors. So they're really dangerous. They're terrible.
Okay, let's thank our speaker.
Thank you.